Hi, my name is Amanda, and I work in Dr. Curtis Thorne's lab, where we study the molecular mechanisms of colon cancer. So when I asked a friend what she thought of when she first heard the word colon, she said, "Oh, I never really knew how to use that punctuation mark." Maybe I'm a little more nerdier than her, but when I first hear the word colon, I think of the underrated organ that plays a vital role in our digestive system. The colonic epithelium consists of many different cell types, and each cell type plays an important role in waste elimination, water absorption, and immune support. The structure of the colonic epithelium is very organized and regulated in structures called crypts and villi. The crypt is located at the base of the structure, and in the crypt lies the stem cells, where they differentiate into specialized cells and travel up the crypt towards the top of the structure, which is known as the villi. The villi is where these specialized cells perform their function. The stem cell in the crypt regularly replenishes the cells up in the villi. Therefore, the colonic epithelium is highly proliferative. Although self-renewal of the cells in the colon is critical, this can be problematic because the more times a cell divides, the probability of mutations increases, which can lead to a cancer phenotype. Colon cancer is the second most deadly cancer in the United States for both men and women, with around 50,000 deaths and 140,000 cases reported in the US in 2019. Colon cancer is highly correlated to obesity and other intestinal bowel diseases, leading to these individuals with increased risk of colon cancer. Over 80% of individuals with colon cancer have developed mutations and proteins in the Wnt pathway. This pathway is vital for regulating the proliferation of the stem cells in the colonic epithelium. APC is the most common protein that is lost in colon cancer, but other proteins like GSK3 can lose their function and promote cancer. GSK3 is a kinase, or a protein that phosphorylates other proteins. Phosphorylation is like a signal, and when this signal is lost, messages cannot be relayed properly. Our lab did an RNAi screen in human colonic epithelial cells, which I'm going to be referring to as HCEC cells. RNAi is a molecular technique that allows us to suppress the translation of a gene product. With this technique, each kinase was silenced and cellular proliferation was measured. The screen showed that few kinases acted as tumor suppressors. When silenced, cellular proliferation increased, indicating that the normal function of these kinases is to suppress proliferation. With these findings, my project blossomed. We selected 20 kinases as experimental targets based on their anti-proliferative function and their novelty in the colon. My project centers on understanding how these novel kinases function in the colonic epithelium. We wanted to see how the knockout or absence of these kinases in the colon affected proliferation, cell morphology, and signaling. My job was to completely eliminate one of the kinases from HCEC cells using a molecular cloning technique called CRISPR-Cas9. To knock out these kinases using CRISPR-Cas9, I used molecular cloning to create a plasmid that had both the Cas9 protein, which would knock out the kinase, and a short piece of DNA that correlates to the kinase of interest. In order for the Cas9 protein to eliminate the kinase from the cell, the plasmid had to contain a sequence of the kinase of interest. This is so that when the Cas9 protein is inside the HCEC cell, it can recognize the kinase RNA and destroy it. Once I created the plasmid and verified that it was correct, it was ready to be transfected into HCEC cells. One of the novel kinases I worked on was called CLIC3, which was a kinase that was the center of my mentor's project. She used my plasmid construct to knock out CLIC3 in HCEC cells. On the left is a regular HCEC cell with CLIC3 still in the cell, where the right is displaying HCEC cells where CLIC3 was knocked out. The images demonstrate morphological differences between wild type and CLIC3, particularly showing that the CLIC3 knockout cells are more elongated as well as displaying a less organized and defined cell shape. We strive to take more images to analyze proliferation and signaling differences between wild type and knockout. And that concludes my talk about some research going on in colon cancer.